This sheet is going to cover prokaryotic and eukaryotic cells. There's a comparison between the two. Prokaryotic cells are very simple, very basic, and then we have eukaryotic cells as well. So I'm going to go kind of quickly through this. I want to keep these videos short. Nobody wants to listen to an hour and a half talk on cells or anything. So I'm going to start with the prokaryotic cell first, this guy. Prokaryotes are bacteria. They're very simple. They have an area right here in the middle called a nucleoid here, and then that contains one long circular chromosome. Bacteria are very simple. They divide pretty quickly. Some divide every 20 minutes, some divide every 30 minutes, and they can adapt to things pretty easily. Fimbriae are these little extensions that come off this way like this. Those are fimbriae. Plasmid, a little circular ring of DNA right here. Plasmids can carry luxury genes, extracellular DNA, and a cell can use that if they're beneficial. We'll get to that later. And inclusion are these little areas here, and they're actually in our model as well from our lab. Inclusions are like a vesicle, but they can hold water or air. They're storage vesicles for bacteria. Escherichia coli or E. coli has the ability to float in water because they store air in their inclusions, so they act like life jackets. Ribosomes, those little red-looking beads, those are involved with protein synthesis, and they basically make proteins, which are enzymes, which for microbes are very important. They rely on enzymes to convert their food energy into ATP. And they can do that with or without oxygen. Some are aerobes. Some bacteria are anaerobes, where oxygen actually kills them. There's a category called a facultative anaerobe, which means they can grow with or without oxygen, so like a switch hitter in baseball. I showed you fimbriae, I believe. Fimbriae are the little extensions that allow a cell to adhere to a surface. They're like Velcro. Neisseria gonorrhea, the sexually transmitted disease, have fimbriae. They stick to the urethra, and that's where they cause problems. This pilus, and sometimes they're called pili or sexual pilus. These can connect to other bacteria cells, and then bacteria can exchange genes through that pilus. Flagella move the entire cell, and you can see where it attaches right there at the bottom. If you get kind of close, let me see if I can get my... here you can see where it anchors in, and then it goes back this way. Now, plasma membrane, same thing, just another way of saying cell membrane. Plasma membrane, you hear that a lot in microbiology. That's the inside layer. Eukaryotic cells have a plasma membrane as well. Cell wall, that's found in bacteria. Some eukaryotic cells, like plants, have a cell wall. That's a second layer. And then this is a capsule, which a capsule, you see this kind of thick, it's also called a glycocalyx. It's a sticky outer layer. Same thing, it allows this bacteria to adhere to a surface. It helps prevent it from being phagocytized, meaning broken down. So bacteria that produce a capsule, a lot of times are pathogenic. Okay, let's go to our lab model. And you can click on these pictures. I try to do this with most of them. Some of them won't let you do this, but this is, you can get far away. This is our prokaryotic cell model, and it's taken from an area that looks like it's up and down. I might post another version of this where it's lengthwise, but here's the nucleoid here. That's the long circular chromosome. If you click on it, the red rings looking things, those are supposed to be inclusions. The blue are ribosomes. And then down here at the bottom, see where the anchor is? There's the flagella coming off. You can actually see up here fimbriae, little yellow extensions. And then the cytoplasm would be everything that fills up on the inside, the jelly material. I don't know if I pointed that out on here. It says cytoplasm. It's just like this jelly material that fills up a cell. All the biochemistry happens there. It's made of cytosol. All right, so that's there. And then if you get really close, here's the plasma or cell membrane, here's the cell wall, and then out here is the capsule. So it's got the 3 layers as well. You can just barely see the capsule on top. So this is the section that's cut in half. But that is everything, and I'll go through the last part with you on the table. So that's everything in a prokaryotic cell. Okay. In 3, 2, 1. All right, this is a eukaryotic cell. Eukaryotes, a category, means true nucleus. They have a nucleus, and that's plants, animals, a whole category of species that have eukaryotic cells. This is not the greatest diagram, but at least it has all the basic parts. That's why I like it. Here's a nucleus, and then that little area in the middle is called a nucleolus. So, and then what you have inside here are chromatin, which are long strands of DNA, and those condense to form chromosomes during cell division. So that's found inside the nucleus. So the nucleus controls all the cell activities. That layer on the outside is called the nuclear envelope. It's a selectively permeable membrane. It keeps holds the nucleus together. It allows certain things to leave the nucleus, excludes other things from getting inside. That's a nuclear envelope. And the pores would be the little openings you see right there. Don't worry about cell coat. That's supposed to be the term for the cell membrane, or plasma membrane. You can see that there. This has to do with a different kind of eukaryotic cell, but plasma membrane's the outside. These could be cilia coming off, little extensions that move substances across the surface of a eukaryotic cell. This area here, with a bunch of tubes and fluid-filled vesicles, is called the endoplasmic reticulum. The endoplasmic reticulum is a series of tubes or elongated vesicles that transport substances around the cell interior. The rough has ribosomes attached, those little dots, so it's got the speckled appearance, so it kind of has a side function. Ribosomes are involved with protein synthesis, so the rough actually has that as a function as well. Down here you can see in the corner, that's called the smooth endoplasmic reticulum because it does not have ribosomes attached. That little area there, there's no ribosomes. There's some free ribosomes. They also float around inside the, well, they're found in the cytoplasm of a cell. Nothing really floats around, but I'm going to back this up. There's some mitochondria, or mitochondria, they're plural. The powerhouse of a cell. They convert food energy into ATP using oxygen. Cytoplasm is the jelly that fills up the inside. It also is an area where biochemistry of the cell takes place. It has amino acids. Here's a lysosome. Those actually break substances down within a cell. Lysosomes have digestive enzymes, and they break down worn cell parts, microbes, food material. There are 2 kinds of white blood cells that we cover in microbiology. One's a monocyte, the other one is a neutrophil. They are phagocytes. They eat other cells. And so lysosomes actually break down once a white cell engulfs a bacteria. The lysosomes then break those down. A Golgi apparatus, this little squashed thing, it actually, by definition, it packages and modifies substances to be secreted out of the cell. It makes little vesicles that come off. A vesicle is something that can move a substance in and out of a cell. Centrioles. Every cell has 2 pairs of centrioles. They're involved with distributing chromosomes during mitosis, or meiosis even. So those are there, and I believe that is everything. That's all the big parts of a eukaryotic cell. Okay, we're going to go to our lab model. This is actually in our biology lab. So there's the nucleus in the middle. Now I know it looks like there's 2 dots. You can click on this. That's a nucleolus. Here's the nuclear membrane. There's the pores. So that's the selectively permeable membrane. Allows things to go into the nucleus, excludes others. Excuse me. Here is the endoplasmic reticulum. This happens to be the rough. It's got the ribosomes attached. Down here is the smooth, the blue. You see there's no ribosomes. This is a mitochondrion. So this cell has 1, 2, 3, 4. It's got a bunch of mitochondria. The yellow here is a lysosome. You remember they break down. They're very important in microbiology because they help break down microbes. The red would be like the free ribosomes that are kind of found in the cytoplasm. They're involved with protein synthesis. The red up here, that's a Golgi apparatus, and you can see if you follow it up, it's making little vesicles that are then releasing something to the outside. So it's like the gift wrapper at Christmas. It packages and modifies proteins or substances to be secreted from the cell. That area right there, there's the centrioles, so the number 12. And I believe that is it. And I showed you the mitochondria. Here's a cell membrane or plasma membrane on the outside. Okay, and I believe that is it. And you can kind of see the cell membrane would just be this outer layer up here. And you can see something's being released at that point. So for the end of this, we'll finish this out. So here's a table. What are the differences between a prokaryotic and eukaryotic cell? Cell wall, yes and yes. Prokaryotic cells do have a cell wall. I showed you that. Human cells do not have a cell wall, but plant cells do. So that's why it's yes and yes. Plasma or cell membrane, yes and yes. We went through that. Nucleus, no, not in a prokaryotic. It's called a nucleoid. Eukaryotic cell, yes, absolutely. Nucleolus, yes. It's a site of ribosome formation. They actually form ribosomes. Nucleoid, no, in a eukaryotic cell, yes, in a prokaryotic cell. Endoplasmic reticulum, yes. These are called membrane-bound organelles, so none of these are going to be found in a prokaryotic cell. Transport substances around the cell. The rough endoplasmic reticulum has ribosomes attached. It has a side function as well. The smooth does not have ribosomes. That is involved with producing lipids or lipid synthesis. Golgi apparatus, yes. Half a dozen flattened sacs. It packages and modifies things to be secreted from the cell, a substance. Vacuoles and vesicles, yes, they're found in eukaryotic cells. They're actually called inclusions in a bacteria cell. Lysosomes, yes, they contain digestive enzymes that break down worn cell parts, microbes, that kind of thing. Mitochondria, yes, they are the powerhouse of a cell. They take food energy and convert that into ATP using oxygen. Cytoskeleton, didn't really cover that. All cells have eukaryotic cells. They have little proteins inside here that act as a cytoskeleton. It's for support, so it gives it some rigid structure. Centrioles, yes. Remember, they distribute chromosomes during cell division. A capsule, no, not in eukaryotic cell. I know that diagram says cell coat. That's not it. They are found in bacteria. Once again, they protect against phagocytosis. Cilia and flagella. Flagella only for bacteria. Eukaryotic cells have both. Cilia move substances across the cell surface, whereas flagella move the entire cell. Human beings have a ciliated layer in the lungs. It's actually called pseudostratified ciliated columnar. It traps dirt and things that are inhaled. Cytoplasm in both, yes. And it is a big area where biochemistry takes place. It holds all the organelles in place. And you do have the pictures here as well. There's our model for the prokaryotic, and here's the one for the eukaryotic. So I hope this video helps, and thanks for joining me.